So, transitioning a little bit from upstream to downstream, we know the argument here is the more lift and effort you put into your upstream, the hopefully, right, the less work your downstream colleagues have to do. Are there innovations taking shape in downstream purification realms from a technology or operational standpoint that you think are going to pave the way for higher quality AAV? Andrew, we can start with you on this one and then transition to Justin.
Oh, so one thing I didn't say with the upstream that I probably should have said is if you can get to a certain level of titer. So a lot of people, when they make AAV, they'll do a solid liquid separation, usually with some kind of filtration step, and then they'll do a tangential flow filtration to concentrate the virus up before they put it on an affinity column, which will specifically bind to the AAV, and then do kind of bulk purification. So concentrate and do a lot of 2 to 3 logs removal of our impurities. So the step there is the TFF, where you concentrate it up. You do that because you have so little AAV that in order to load it on an affinity column, you would need to load it for several hours, days, too long. So you need to concentrate it before. So if you can get the first step, I think a lot of the companies are at now, is if you can get your productivity of your upstream high enough, you can just completely delete that TFF. I call it TFF1, that first concentration step prior to affinity. And that's the whole. So if you imagine the yield of any step is like 80–90%, I mean, with AAV, then that's an instant bonus in yield and time. So I think that's a good thing to strive for with the purification. Other than that, there's different affinity resins coming out. I've been a proponent for maybe affinity membrane chromatography. So you can just churn, because we don't really care about capacity so much at this point. It's more being able to process large volumes quickly. So I think that's kind of a need that could be met. And then there's a lot of development with the empty removals with anion exchange. There's both innovator improvements and then the vendors. For example, Sartorius brought beer separations, and they've recently come up with a more reproducible version of their SimQA common use SimQA column. So I'll probably go to Justin now before I go on too long, but there's
Justin.
A lot going on.
Well, what catches your eye?
I think you've hit on a number of opportunities in there. I mean, elimination of process steps always should have the benefit of reducing losses in the downstream operation. I think a lot of this in the industry has gone this, where they found the ability of empty-full separation chromatographically has really, the understanding of that and the ability that's really improved over the years, being able to transition away from ultra-centrifugation based process that you may have seen in some of the earlier ones coming. So I think, newer resins are becoming more available, techniques are becoming more available towards it, so that you can continue to innovate around this. And I think ultimately in a more scalable format and looking more like a traditional biologic process here.